We will start with part 4 of this lecture of the workshop, Gender in Research, with Sabine Oertelt-Prigione. And she will pick up the conceptualization of sex and gender again, and now for journals and grants.
Well, welcome to the last of our 4 lectures together. And this time I'm going to talk about, pick up again, if you want, the operationalization of sex and gender, but tailor that a little bit more towards journal and grant writing. So what does that mean for you when you write a grant, when you write a paper for a journal? What should you take into consideration? And how can you present that in a way that it reflects what we've been trying to teach you in this summer school? Okay, so we'll start again, just pick up some of the concepts of the operationalization on sex, but bring that a little bit more into the practice and potential things that you might want to address when reporting your studies. Then when it comes to the operationalization of gender, how could that be done? What should you be reporting and how to do that? Then we'll briefly look at what granting agencies want you to do and why they want you to do that, and what does that mean for the practice of grant writing. And last, if you report about sex and gender differences in your publication, or actually you just include information about sex and gender in your publications, how should you ideally be doing that, and at what levels in a manuscript should that be reported? All right, we discussed already in the first lecture that there's basically three levels that we can use to define sex. So we can analyze genes, we can analyze hormones, we can analyze external genitalia. And then the question you will have to be asking yourself now is, well, once you approach your experiments and you approach the research you'll be doing, which kind of measure will you be using? And then ideally, give some details about your choice. So why are you deciding to measure hormones, and when are you deciding to measure these hormones? And in which way could that be relevant to the work you're doing? Or why do you not need to do that? Or, pick information about genetics, for example. Ideally, there could also be a combination between the methods you're applying and the type of analysis you're doing just for sex. However, that doesn't necessarily have to be addressed. Then, of course, the question also is, are there any variations that are being considered? So are you working with a dichotomous, with a binary concept, or are you actually including variations thereof? So if you use external genitalia as criterion, will you also include, for example, children that have undefined or not clearly defined or attributable genitalia to either female or male? Will you be working with hormonal concentrations, and how will you allocate those? Which kind of cutoffs will you be using? Will these be binary? Will you have intermediate categories? And if you work with genetics, do you consider anything that goes beyond XX and XY? And if you don't, why? And why not? And furthermore, another concept could also be, which kind of terminology will you be using? For example, will you be using differences of sex development? Will you be using intersex? Will you not be addressing that, and so forth? So think about also the kind of terminology you would like to use, and also if that has some kind of meaning for both the readership, but potentially also a larger community of patients or people that you're addressing with your research. Then let's move on to animal models. So when it comes to the animal models, for the longest time, we have assumed that we pretty much can regulate anything that animals are exposed to. And so we can control anything. We can regulate how many are in a cage, what they're eating, how the temperature is, how much light they're exposed to, and so forth. And then it turns out that actually these things we're controlling might have a different impact on female or male animals. So for example, the social dynamics in the cages might be different if you only house females or males, and depending how many those are. What you're giving them in terms of diet might have an impact. So for example, soy-based chow is estrogen mimetic, so it's a phytoestrogen. If you are putting them in a room with different sounds or different temperatures or different rates of change of these, they might react differently to stress. And last but not least, the research staff handling the animals might have an impact on the reaction of the animal. And that's most important, I would say, if you look at any reactive behavior of the animals, a little bit more than if, for example, you're performing a sham operation or you're performing an operation on the animal. So it depends a little bit. Are you investigating the behavior or are you doing an operation on the animal? So the potential factors that you see with animal research can also apply to cell cultures. So as I said initially when we were talking about the operationalization of sex, we don't have a lot of information about our karyotypes. I don't know how many of you actually know their karyotype. And the same thing, unfortunately, goes for a lot of the cell cultures we're using. So very frequently I ask rooms in which I do these lectures, well, do you actually know the sex of your cells that you're using in your experiments? And there's always people who don't know. And sometimes it's a large group of people that don't know. The second step here is the stability of the karyotype. So we know that, for example, in tumor cells, over time and through passages, the karyotype, the makeup of the chromosomes will change. So they can lose certain chromosomes or they can actually duplicate or triplicate certain chromosomes. And the same thing applies for the X and the Y chromosome as well. So especially if you're working with a tumor cell line that has been in culture for very long, the karyotype today might not be the same as it was a few years ago. And it might be worthwhile to check that every once in a while, not just for the X and the Y chromosome, but in general. If you are doing some kind of changes with your cells, so for example, you're reprogramming them into pluripotent stem cells, the process itself of reprogramming changes the methylation patterns and changes the inactivation patterns of the X chromosome. So usually one of the two X chromosomes is inactivated and only about 15 to 20% of the genes of that inactivated chromosome are still transcribed. But when you go through a process of reprogramming and then programming again, so let's say you go through the process of making a stem cell or a pluripotent cell and then differentiate that again, going through a demethylation and methylation process might actually change the methylation patterns on the X chromosome. We don't know exactly how much media influence all of our cell cultures from a sex-specific perspective, but we know that, for example, certain coloring agents, phenol red, have an estrogen mimetic effect, and we know that certain types of cells seem to be growing better, female cells seem to be growing better than male cells in culture. We don't exactly know for all of our growing media. And of course, the other thing to take into consideration is the potential methylation differences depending on the cells of origin, because if you are taking blood from a participant in a study, those cells, even if you clean them, you wash them a few times, they have been swimming in a hormonal environment up until a few minutes or hours ago. And of course, that has an impact on methylation patterns possibly. What we don't really know yet is if different estrogens in culture have different effects. We know from human studies, so for example, the Women's Health Initiative, that different types of estrogens, so be it synthetic ones or purified ones, did have a different effect. So in the early phases of the Women's Health Initiative, the estrogens used were purified equine estrogens, and in the later phases were synthetic estrogens, and they have a slightly different effect and definitely a different degree of purity. We don't know if that also affects cell in the Petri dish. And the other effects, as I just said, were also media. We are using all kinds of different media in the lab, and we don't really know if there are intrinsic growth differences between the cells, depending on the media we use. I briefly touched upon the examiner sex before, and that's something that was quite a paradigm shift a few years ago. We always thought that even if we cannot control everything, as I said before, there are certain factors that might influence still in a sex-specific manner. The experimenter doing the experiments shouldn't be one of those, because they're basically two different species. So the experimenters are humans, we're handling rats or mice, there should not be much of an influence. Well, fast forward to the year 2014 when this report came out, where in the lab of Jeff Mogul in Canada that studies pain and response to pain and response to painkillers, they have found that depending on who the investigator was, the animals, the rats in this case they were studying, were responding differently. And so especially the female mice, if the experimenter was male, so if they were smelling male pheromones, reacted differently than they would to female pheromones. And if you want, interesting and scary part of that is that really there seems to be a crossing of an interspecies barrier in the effect the pheromones can have. Because as long as we put the same animals in a cage, we can predict their reactions. But now it seemed that these animals are actually smelling the pheromones of whoever is handling them. What does that mean in practice? Well, it has been reproduced in a few other labs, always in the field of pain research. But it is definitely something that we might have to look more in depth into in the future. And the first step towards mitigation might be at least to report who is doing these experiments, or if there has even been some thought given to who is doing these experiments, and potentially report that in the methods of our papers. If you want to go a little bit more in detail, I showed you the website of Gendered Innovations in the first lecture, and we have reorganized several of the general analyzing methods, and you will see that if you go to analyzing sex, there is this almost circle with all the different steps of the research process. And you will find recommendation and references to methods, specific methods and case studies for all the single steps of the research process. So I would advise that you also go there. And if we haven't covered, or if basic research is not covering what you're doing, you might find a lot more information and a lot more in-depth methods on how to analyze that also in different fields. So what should be the essential steps when you report your analysis considering sex? Well, first of all, we would like to know the rationale of the study. So why are you doing this really? Why are you analyzing sex, or why are you not analyzing sex? The second step should be, we would like to know how the experiment is set up. So how many subjects or cells or animals are being used, and please let us know the sex of these participants or tissues. In most cases with human subjects, we do know. In animals, we sometimes know, and in cells, we mostly don't know. So there's definitely room for improvement here in trying to make that more transparent. All potential confounders should ideally be listed. As I said before, if you're working with animals or with cells, you could give us a lot more information about the housing condition, the chow they're eating, the light and dark, the temperature, and possibly consider if it makes sense to report data on who's conducting these experiments. Consider if there are any potential interactions, for example, with age, and in case, analyze that, and of course, analyze by sex. And then the crucial step, and I underline that here, and it will happen again later, please report them in a disaggregated manner. And this is where actually a lot of the issues are happening. People do report how many animals have been included that are female or male. They even do the analysis, but when it comes to the reporting, we rarely see disaggregated data. However, in the future, if I want to go back and do a meta-analysis, I need that disaggregated data in order to put together several different experiments and have much more robust data. So please report that in a disaggregated manner. And if you don't do it in the paper, you always have supplementary methods to do that. And then it would be nice if you discuss the implications of what you've just found. So much for the reporting of sex and what we would like to see. Let's move over to gender. As I said before, the complexity of gender is really to define what it is you're investigating. So are you using a gender index, and then why and which one are you using? But if you're not using a gender index and you want to investigate gender, give us some information about which layer, which dimension of gender you want to investigate. Maybe you only are going to investigate identity. Maybe you will investigate gender roles. Maybe you plan to investigate gender relationships and so forth. And then please let us know why. Again, also here, We also redeveloped a complete new set that shows how gender can be analyzed in different types of research in different fields. Again, here I would advise that you go to the Gendered Innovations website, and you again will find a circle with the different phases of the research process, and you will find information and methods for all those different phases in different fields of research. The reporting of gender, again, I showed you some questionnaires before. The reporting of gender follows a bit along the lines of sex, but of course with an added degree of complexity, I would say. And here is what Petra Verdonk mentioned in one of our discussions before. Did you use a theoretical framework? And if yes, which one did you use? So it would be really nice if you didn't only give a rationale of what you're doing and why you're doing this, but also said something about the conceptualization of gender that went into your study. So why did you choose to take a certain dimension to investigate that one and not another one? Which kind of theoretical background is informing the work you're doing? Where does this come from? Has there been previous work in that direction that you can build on? Then we would like to know, how are you operationalizing this? Are you using questionnaires that are existent? Which ones are you using? Why are you using those? And how could that possibly compare? What you find can possibly compare to those? And if you decide not to use any of those and you maybe developed your own, why did you have to do that? Again, of course, it would be nice to have an experimental setup table with the number of subjects participating. Depending on the dimensions of gender that you consider, give us this breakdown as well. So are you using identity, then show a breakdown by gender identity. If you're using any kind of measure for gender roles, then show a breakdown of those gender role categories and so forth. Are you applying an intersectional lens? If you do, please let us know why and in which kind of form are you including this intersectional lens. It is not always easy, and especially not if you're doing clinical work to include that. So let us know what you're doing. Are you doing a subgroup analysis? Are you doing a stratified analysis by several dimensions? Which kind of intersectional questions are you addressing and which ones not? Again, the most important part, please show your data in a disaggregated manner so that others might afterwards be able to go back. And I cannot stress this enough because this is really the biggest lack we're seeing today is a lack of disaggregated data, which would make some of our work much easier and could actually allow us to go forward faster. And of course, the implications of the work you've done should be discussed. Now, why does this matter? Apart from the fact that it is, of course, for all of us trying to teach in this summer school, we all consider that this is really important and you should be doing this anyway. Granting agencies all over the world are also asking you to pay attention to sex and gender in your research. And I'll try to give you a brief overview of why they're doing this and what that means for the principles of research you're working with. So you will see that depending on where you come from, and I only picked a few of these, sex and gender are not only conceptualized differently, but sometimes priorities are set in different ways. So if you work in the United States, a lot of focus in the past has been placed on SABV, so sex as a biological variable, much more than on gender. And the attention towards the intersection between the two and gender itself only started growing in the last few years. So gender was allocated much more in the gender studies and not really that much in biomedicine. In Canada, there is a tradition to look at both sex and gender, and of course, you will know your slogan of every cell as a sex and every body as a gender, and this has really informed the, I would say, Canadian approach to this over the years in bringing in both concepts. And in Europe, we mostly talk about the gender dimension when it comes to the incorporation of sex and gender into research. And this is a definition that comes from the European Commission, not from a single country, but several countries are actually picking this up or adapting it to what their own formats are. I don't expect you to read all of this, or I definitely will not read the whole text to you, but I wanted to show you, and you can look at those later on when you have time, the rationales of why these agencies say that it is important to consider sex and gender. And this is the one from the NIH, which SABV. And what they say is the policy is meant to be a new standard to enhance reproducibility through rigor and transparency and increase the knowledge base. So this is why the NIH felt that it is important to address and to consider sex as a biological variable. If you look at the Canadian Institutes of Health Research, so this is the statement from the Institute of Gender and Health, it is about improving the rigor, reproducibility, and generalizability. It's about excellence. And when you go to Horizon 2020, we are now since a few months under the umbrella of Horizon Europe, but not everything has been updated yet. And this statement will stay the same. It will actually be incremental. There will be a few additions to it. But what the European Commission says that considering the gender dimension will improve the scientific quality and societal relevance of the produced knowledge, technology, or innovation. So overall, if we bring all of these together, what does it mean to look at the gender dimension or sex and gender in research? Basically, what they all have in common is they say it has an impact on the reproducibility of your work. So you're offering more information, you make your work more transparent, which makes it more easily to reproduce. You will produce more excellent work because the degree of complexity and the degree of information you're offering might increase the quality overall of your work. And it will definitely increase the societal value because the more diversity you include in your work already, the more representative this might be of society at large. So overall, it's some big expectations that funding agencies are placing on why this is important. One special thing about the European Commission, so in Europe, it is not just about the incorporation of the content. So integrating is the third one of these, integrating gender and sex analysis in research and innovation. But there are also two other layers that are being addressed. And one is the fostering of gender balance in Horizon 2020 or in Horizon Europe research teams, and the ensuring of gender balance in decision making. So what the EU is really doing here is it's kind of splitting the whole thing into two layers to make evident that there is a connection between context and content. So content is what we've been mostly talking about in the summer school. How do you actually do the practice of research? However, the context, so who is doing the research and with what opportunity and with what power in one way or another influences the kind of content that is being done. So we all know that as researchers, we're human beings, and as objective that we want to be, we are influenced by our life experience. This influences what we see, and it influences the questions we ask. And so the context and who gets to do research also shapes the kind of content that we will be producing. What does that mean for reporting upon publication? So let's say you have to write a paper, and how do you want to report that? So this is a very basic, and if you want to do into any formal bibliographical research, please don't repeat that at home. It is just a really simple way of showing you how far and how often the terms sex and gender have been included in publications in PubMed in the last decades. And you really see that there has been an incremental change since around the year 2000, and the numbers are going up, and there's definitely more and more attention being paid to this and more reporting that includes sex and gender as variables in research. What does that mean? That a lot more research is being reported that takes sex and gender into consideration, but that also means that we should have some kind of guideline that lets us know what to report. And I've already told you some things before, but now in a formalized format, I recommend that you have a look at the SAGER guidelines, and SAGER stands for Sex and Gender Equity in Research. What you will find is two tables. The first one basically shows you where to include this, and what they're asking for in the paper is you offer a rationale, you have a clear understanding of what the concepts of sex and gender are, which ones are you using and why, and then what they're showing you is at what layers of the paper should you be saying what. So of course in the introduction it would be nice to know about theory and rationale. In the methods we want the detail about what you've been doing, why you've been doing it, and what has been included. In the results, please disaggregate your analysis. And of course, in the discussion, let us know what you learned from all of this, but placed together in one table. There is also a very neat figure for editors in trying to make clear what you should be looking for when papers are being submitted for publication. Several large publishing houses are requesting that you give information about sex and gender. So the Springer Nature, at least to a certain degree, most of the Nature papers want it, Science wants it, the Cell family wants it, the Lancet family. They've all published big statements saying that from now on, you need to include an attention to sex and gender. You need to report that. Some are following through more strictly than others, but in general, that's probably also one of the reasons why we're seeing this increase in publications. Overall, what it basically says is sex and gender relevant for the topic of the study? And if it is, editors should be checking if there is information about sex of the data acquired, if sex has been considered in the design of the study, and if it is addressed in the discussion, and possibly if it represents a limitation not having been addressed or in any other form. So if some of these three questions are answered with no, the recommendation is to send it back to the authors so that this can be added before it actually goes out for peer review. Now, my last recommendation for steps towards mitigation overall. Obtain as much information as you can about your samples. And this goes for both cell samples, but of course it also goes about populations you might be including in your studies. Try to understand who you're working with, under which conditions, what access do they have, and so forth. And if you're working with samples, with biomedical samples, try to know as much as possible about the sample itself and where it came from. Include at least both sexes in your study, unless, for example, you study prostate cancer, and then it might make sense to only include male cells. However, this is an exception. In most cases, it makes sense to include at least both sexes. And think about how you want to conceptualize gender and how you want to measure this. It is helpful to consult experts. So there are more and more experts out there, and some of them are really based in a specific discipline and can really help you dive deeply into this kind of work in specific disciplines. So when you are starting out, look out for experts in your field that can support you and that can give you feedback on the work you're doing. Always disaggregate data, so that's mandatory. Always stratify your analysis by sex and gender. It's always mandatory. And of course, that's the most important of all. Report your data in a disaggregated format so that other people can find it. And now that you are in the summer school for a while, and you will still be continuing for a little bit, you have a mandate to also spread the word about this and share your knowledge and try to involve colleagues in a discussion of why this might be meaningful and why it might improve their research and why to pick this up. And with that, I thank you very much for your attention and look forward to your questions.
So the last discussion now about part 4. Petra, when I hear the lecture of Sabine, I already said to myself, well, if you use the Sager, then it is institutionalized in your department properly, sex and gender. Don't you think so?
Just using the guidelines, yeah. No, they're really great. I mean, it's wonderful that they're there. But then getting people to use the guidelines, that's basically, that's the next level implementation.
Yeah, that's not very easy?
No, because for those who use the guidelines, first this step towards awareness and towards implementing in their design, etc., has to be there. So if you find it important, that's when you start using the guidelines. And then it's like a checklist, whether did I do everything correctly. Did you use it yourself? I do, yes, I do use guidelines. I also do, I don't do very much clinical research and like only occasionally. So for my type of research, these guidelines are not really applicable. But I do gender research like all the time.
Yeah. And do you use them, Marijn?
Well, and one of the things that came out of my participation in this workshop last time around was that I could only dream Sager for weeks at a time after that, of course. But also we were tasked with making an implementation about how to share this knowledge with our wider, our colleagues, but also social circles maybe, to promote this uptick, right, in the wider academic field. Because we can be evangelical about it, but the knowledge needs to spread. And
So what did you do?
well, I'm glad you asked, because we made a game about this. It's
A
called
game?
A game, yes, funded by ZonMw. It's a sort of well, you're talking to younger people here, right? Slightly younger people who are maybe for the first time trying to think about these concepts. So we thought, how to reach them? Let's make a game that is easily playable in a lunch break or something like that. 30 minutes. It's called Gender Quest, the quest for better research. And if you play it, I think there's
I
a
like
link
the
here
underline already.
Yes, it's better research, right? So it's designed for scientists that are thinking about their next research, how to design it, or PIs that are trying to steer a research line, but also people involved in policy or in grant advising. And it really takes all these steps that were outlined in the Sager guidelines and by Sabine
So
design.
you evolved Sager into a game.
Exactly, and and you go through it. You go through all the steps in a slightly gamified fashion, and in the end, you might reach the golden grant, and this will be your reward.
Okay, interesting. But when I hear you talking in your lecture, I already said to myself, it's the why, the how, and what. So it's the Simon Sinek circle, which you have to do to make a good conceptualization of sex and gender. That sounds very easy, but why is it so hard?
Because people have diverging priorities. I would put it like this. So the Sager guidelines are not at the same level as, let's say, Prisma, guidelines or other kinds of guidelines for qualitative research or quantitative research. We have all these kind of nice checklists where you have to put down everything, and the Sager guidelines are not up there yet. So they should possibly be there as well, and just be given more of that kind of stamp of approval of being just a mandatory thing. There are some journalists who are actually pointing to them and ask that you
because
kind of
I think it's very interesting that publishers already have guidelines to say yes or no if it comes to gender and sex. I think that's profitable, isn't it?
It is. The point is, as I said, there's conflicting priorities. And as I said before, what sometimes happens with grant as well is, a reviewer might read something or an editor might read it and say, well, they didn't do sex and gender sensitive research, but it is wonderful and innovative, and a lot of people are going to read it. The altimetric score is going to go through the roof, and we love that. So for this one,
so
we don't
it isn't
care.
mandatory.
And that's, we have to face the fact that in publishing, there's also an element of novelty, of innovation, of, PR that you're going to create. And we all try to do the right thing. And I would say most publishers try to do the right thing, but there's also other factors that are conditional to the survival of researchers and publishers.
Yeah.
If I can add to that, maybe, because I think that there's a trend towards also post-publication peer review much more these days, with articles being put online in preprint servers and then people just engaging in that kind of collective peer review. So there actually might be an opportunity to just type a comment. So what about Sager, and then see how people react to that.
Yeah.
Rather than having a gatekeeper function with the editors.
I'll employ a student who just spends their days and going through all those fora.
Yeah. I was amazed that even the sex of a examiner changes the result of a research. Did you know that?
Well, I was also flabbergasted when this came out, and it's I guess not completely unexpected, because we know that there are experimental effects between different people who are handling animals in our labs, and we usually attribute it to experience or some other factor. But yeah.
So we have to take sex and gender into account.
We do, or find a way that we don't exude pheromones anymore. But I'm not.
Can I add to that?
Yeah.
Because I think it's not unexpected, as Marijn says, but the idea that animals have no social life, I think that's a weird idea, right? They're living in a social world as well, and there is learning in animals, and there's also social learning, and they teach next generations, as social biologists already say for years. So there's also social learning,
and
social
even memory
life.
is memorized in genes. Yeah,
And that's the
in the
methylation,
genetics. Yeah.
the methylation discussion Sabine referred to, and that's transferred to next generations too. So that ends up in germlines.
Yeah.
So there is no social without the biology. There's no biology without the social. And the idea that you can distinguish the two neatly, that's been a mistake, I think, for a long time.
Yeah. Peter, Sabine mentioned all the steps you have to take into account, especially the disaggregated data, for instance. What do you see often when your PhDs, for instance, want to publish? Which do they miss steps then?
Well, we always miss steps. Like, we always could have done things better. And a good reviewer gives good feedback. And we're very grateful, because that's a lot of work. We review ourselves. So yes, there are always steps that you could have done better. In our field, we work with theory, and we design our studies based on theory and a decent, let's say, design from a gender perspective. So we do miss a lot of things, but maybe not those issues in our research. I think when you're socialized as a researcher in a biomedical field where this is a completely different paradigm, then that's maybe more difficult. I really think that this step towards this type of thinking, which is transformative, I think also for researchers themselves, but I do think that that's a major issue. Once you start doing it, it's not that easy. I mean, this is not rocket science. It really isn't. It's quite easy. And we should explore more. We just have to start doing things and report, and we need cross tables. At least we need cross tables. That would be the least thing that researchers could report on.
Yeah. Sabine, you mentioned all those steps, but I think those steps are, common for every type of scientific research, isn't it?
Yes, and the fact that I still have to spell that out at so many presentations that I give makes clear that we really don't see it. We're doing some work right now where we're really looking into the detail of papers where people claim to find sex differences or gender differences, and we're actually looking at what they report and where. And even in papers that actually address that, so let's say not something where spuriously they report it, but really where the goal is or where the reporting is that, you still have huge lacks of information about what kind of methods have they been exactly using. You know, you probably know how many cells or individuals or whatever have been included, but the methods are incomplete, the tables are incomplete, we don't see disaggregated data. The rationale a lot of times is like, really? Yeah, we did it because, and you think, well, was that kind of an afterthought because you did a lot of analysis and that was the only one that was significant? So, it's not a given, and it should be. That's why I break it down in such simple terms, because I really hope that a lot of times my colleagues see that and think, oh, that's so simple. It's almost embarrassingly simple, and yet we're not doing it.
Yeah, but Sabine emphasizes the disaggregated data just to compare the studies and all the steps that are necessary. Don't you think that if we do that very properly, sex and gender aren't just a tide or a wave in sciences, but really get more respect of other scientists, and then it's more easy to institutionalize sex and gender in research?
Well, I think you're going to get better research overall. So indeed, open reporting of data sets is very important, and that's easier now than ever before. The same goes for code. So there is this whole initiative to publish your code with your paper. So basically people can just follow along and see whether you've sneakily omitted stuff in the middle or not. And pre-registration as well. So you want to be specific about a certain hypothesis. Okay, put it up front, put it out there, and we can check the results.
Yeah.
Yeah.
So again, transparency in research is so necessary to have good research. Yeah. Anything to add?
No, I think that's a perfect word to describe what we do, how we do it, why, in our papers. We need a higher word limit in papers. I would recommend that for like
A higher word limit?
well, we have a word count generally for journals, and we have to describe more. So then need to increase the word limit, and then we can start describing all these issues.
Yeah. Agree?
Yes, I mean, it's a well-known meme that people refer to the methods from a previous paper, which then refer to the methods of a previous paper. And then you go all the way back down the chain, and it turns out there's nothing there in the beginning. And that's a problem that we need to fix. So it's really not that difficult indeed. And it should be easy and straightforward to just report your data, just report your methods, and then everything is transparent.
Okay. Thank you so much and thank you for all your lectures today.
Well, thanks for the invitation.
Petra Verdonk, Marijn van Wingerden, Sabine, thank you so much.